Dear students, welcome to Tumula online classes. So today also we'll deal with the next questions of level 1, that is KCET level questions. So let us start with the first question. Middle lamella is composed mainly of. What is middle lamella made up of? In theory we have studied. So regarding this middle lamella, it is a common cementing layer which is lying between two adjacent cells, which is made up of calcium, magnesium, pectate itself, right? So that's why, what is the answer? Let us see here. So hemicellulose he has given. Of course it is also one of the component so of the cell wall, but not the middle lamella. So it is calcium pectate. So this is the correct answer for this question. Next one is muranic acid. Murane itself is the component of the cell wall of bacteria, and phosphoglycerides are not, right? So then the correct answer is calcium pectate. And the next question. So which of the following is enucleated cell? Enucleated in the sense that so there is no nucleus at all in this cell. So companion cell, of course, it is one of the cells of phloem tissue, right? So it has its own what? So nucleus. Pseudotube cell. So this is the enucleated cell in the plants, right? So that is pseudotube cell. So it is enucleated cell, right? Next one, tracheids as well as vessels. So xylem vessels as well as tracheids are dead cells, but what happens here in these cases is initially these cells have their own nucleus, but when they reach the maturity, that moment they lose so the nucleus as well as cytoplasm and become dead vacant cells itself, and such kind of cells are tracheids as well as vessels. Tracheids are usually seen in case of gymnosperms, that is flower so plants, and in higher plants usually vessels are the so xylem vessels are the conducting cells present in higher plants, right? So this is the correct answer for this question. B is the correct answer. Next question. In which region of the interphase chromosome does transcription take place, right? So this is more important question to be discussed over here. What is that? In which region of the interphase chromosome does transcription take place, right? So during interphase, already I have explained, cell division take place in two stages. One is called as interphase, and second one is very important one, that is M phase. M phase in the sense that mitotic phase. So interphase itself is the preparatory stage so of the cell division. Here the cell doesn't divide itself. So after this stage only M phase occurs. In M phase there are four stages that we come across over here. Those four stages are, so what are those one? Prophase, metaphase, anaphase, and telophase. So these are the four stages that we come across. After this one, cytokinesis. So here in these stages the division of the nucleus takes place. So karyokinesis, this one is the first stage, so that is karyokinesis. Under karyokinesis, so what happens? Four stages we come across. So in these four stages the nucleus divides of the cell, and later the division of the entire cell that take place, that is nothing but cell organelles as well as cytoplasm is equally shared between two daughter cells, right? So this is the cell cycle that we must know basically, right? So we are focusing on this interphase now. What is that? Interphase. In interphase what happens? There we come across G1, S as well as G2 phases. Right, in the next chapter we are going to study regarding all these one, so in detailed manner, right? So in this interface, so which is the chromatin material, so that help in transcription is the major concept of this question, right? So let us see, in interface what happens inside the nucleus, there the chromatin material is present, it is lying inside the nucleus, and somewhere it is highly coiled structure and somewhere it is loosely coiled structure, right? So I have already cleared this one. So highly coiled and darkly stained area itself is said to be as heterochromatin. What it is called as heterochromatin, right? So there is no chance of transcription. It is transcriptionally inactive in nature, already have told you. But loosely arranged one is called as euchromatin, where there is a possibility of the separation of two strands of the DNA in between that one. So there the synthesis of mRNA may take place, and that biosynthesis of mRNA on one of the strands of DNA itself is called as a transcription. Hence, euchromatin itself is transcriptionally active in nature. So that's why, so usually, so what is the question? Let us see here once again, in which region of the interface, so chromosomes so transcription take place. Transcription that take place in euchromatin itself. Hence, the correct answer for this question is euchromatin, right? So in interface, transcription occurs in euchromatin, not in heterochromatin. Telomere is nothing but it is the terminal part of the chromosome, where so the chromatin material is tightly or it is a highly coiled structure and darkly stained structure. So that area is also not possible to transcribe there itself, right? So that's why this is also not the correct answer. So centromere, there also the same thing. So that's why, so correct answer for this question is euchromatin. And let us go to the next question. The cell organelle associated with intracellular digestion of macromolecules is, so those cell organelles which have the digesting enzymes within themselves. So already we know one of the cell organelles which is single membrane bound cell organelles, and it is very smaller enough and it is formed from the vesicles of endoplasmic reticulum, and after that one it comes to the Golgi complex. Later it comes, it exists as if the independent cell organelle, that is nothing but lysosome. Lysosome consisting of 40 more varieties of what? So intercellular digesting enzymes, that is hydrolytic enzymes, that is the why. So these lysosomes are the cell organelles which help in intracellular digestion, and they contain the digestive enzymes within themselves. So glyoxysome is there, polysome is there, dictyosomes are there. Glyoxysome is also single membrane bound cell organelles coming under the heading microbodies, and polysomes are those ribosomes which join each other, so binding to the same or a single mRNA, and they form polysome or polyribosome. These are called as usually present inside the bacterial cell. Dictyosomes are nothing but Golgi complex. In case of plant cells as well as lower cells, so they are said as dictyosomes since they are distributed, right? So this is the question to which the lysosome is the correct answer for this question. Next question is, chromosomes can be stained with one of the following chemicals. What chemicals do we use so as to stain the chromosomes? That is nothing but the stains. Some of the stains like acetocarmine, acetoorsin are the stains used for the staining the chromosomes. That's why option D itself is acetocarmine. So which is given over there, it is the right answer for this question. And other chemical chemicals you see there yourself, light, so green and eosin, saffron, these are usually used so as to stain the sections of the stem or something else, right? So that's why, so these are not the correct answer. So acetylcarmine is the right answer for this question. Next question is, respiratory enzymes are present in which of the following organelles? So respiratory enzymes are responsible for the respiration, that is intracellular respiration. One of the cell organelles consisting of these respiratory cell organelles, so respiratory enzymes, and that respiratory enzyme which help in performing intracellular respiration. So one of the cell organelle which help for that one is mitochondria. That's why mitochondria mainly consisting of these respiratory enzymes within that one, so which help in intracellular respiration. That's why mitochondria is the correct answer for this question. So what about the chloroplast? It doesn't help in respiration. It helps for what? Photosynthesis, since it consisting of photosynthesizing pigments within it, right? Lysosomes are called as suicide bags of the cell, and peroxisomes are one kind of microbodies. They help in different functions, right? So a polypeptide is assembled on. See here, next question. A polypeptide is assembled on, right? So polypeptide meaning I have already cleared for you people. So polypeptide is nothing but chain of amino acids, right? See here, so this is one amino acid, so this is another amino acid, this is another amino acid. So all these amino acids are bonded with the help of a bond called as peptide bond, and this peptide bond makes a chain of a peptide, that is nothing but polypeptide chain it is called as, that is nothing but it is going to form one protein molecule. It is going to form one protein molecule. So then, where the protein molecule assembled on, which cell organelle it is confined to? So it is nothing but ribosomes. Simply ribosomes are called as, since ribosomes are called as the protein factories, hence they are assembled on ribosome only, not DNA, RNA, nucleolus, right? Nucleolus itself is called as a ribosome factory, where the synthesis of many number of ribosomes that take place over there. So that's why ribosome itself is the correct answer for this question. Next one. So one of the chief functions of SER, that is smooth endoplasmic reticulum, what it helps for, some of the functions they have mentioned here in options. So let us choose the correct answer over here, right? What is the first option? That is protein synthesis. Protein synthesis, as already I have mentioned, ribosomes are the protein factories, they perform this action. That's why this is not the function of smooth endoplasmic reticulum. But one thing we must remember, rough endoplasmic reticulum indirectly, so it involves in protein synthesis, but not smooth endoplasmic reticulum. So that's why this is not the correct answer for this question. Enzyme production. So it is not meant for any enzyme production itself, and it is meant especially for the lipid production or lipid synthesis. That's why lipid synthesis is the correct answer for this question. What about the fourth option? See here, microtubule production. Microtubule production is done by one of the cell organelles called as centrioles, right? They are responsible for production of microtubules, which forms the spindle fibers during cell division. Many a times I have explained this one during theory, right? So that's why correct answer for this question is lipid synthesis. Next question. Cells divide and new cells are formed from pre-existing cells. The concept was given by, who explained this concept? That is, so cellula is cellula. You know this concept. So means omnis cellula is the sentence said by Rudolf Virchow is the name of the scientist, the one who brought about a new concept in cell theory itself. Cell theory was basically proposed by Sheldon and Scaven, but whenever he studied, means Rudolf Virchow studied the same cell, so that moment he said that new cells are formed from the pre-existing cells, right? So this is nothing but omnis cellula e cellula, this sentence meaning itself. That's why the correct answer for this question is Rudolf Virchow is the name of the scientist, the one who made one revolution in cell theory, right? So next question is, mitochondria are semi-autonomous as they possess. So many a times mitochondria as well as chloroplast are the semi-autonomous cell organelles. I have already mentioned there. Why they are called as semi-autonomous? Why? Because they independently divide themselves irrespective of the cell division itself, so that do not depend on the cell cycle or cell division itself. They independently divide themselves, multiply themselves so as to form many number of mitochondria on their own. Why? Because they have their own DNA. There is one circular DNA is provided inside these mitochondria as well as chloroplast. That's why they do not depend on the genetical material of the entire cell. They have their own genetical material. That's why cell, as if the cell cycle is taking place, so mitochondria division also taking place, but irrespective of the cell division. That's why they are said to be as semi-autonomous cell organelles. Not only DNA, since DNA is there, on that DNA strand, one of the strands of DNA, the biosynthesis of mRNA also take place. That is nothing but RNA is also present there. RNA is also required there. RNA is also present inside the same mitochondria, and even the protein synthesis also take place according to the message what is present inside the mRNA, right? So that is done by the ribosomes. 70S ribosomes are usually present inside the mitochondrial matrix, and these help in synthesize the proteins required for the construction of this mitochondria. That is why so these mitochondria are said to be semi-autonomous in nature, since they consisting of DNA, RNA as well as ribosomes. Hence option C is the right answer for this question. Okay, we shall go to the next question. What is the next question? So very interesting one. Some of the statements are given over here. 4 statements are given over here, and there are among them which are correct, which are wrong. We'll have to analyze over here. Let us read the statements first. Plant cells have centrioles. Already you know, plant cells do not have any centrioles. Centrioles are confined only to the animal cells, but in lower plants, so these centrioles are present. Already I have mentioned that one, but since he has given only the plant cell, so majority of the time plant cell do not have centrioles. That's why initially the statement says the wrong statement itself. That's why option A is not the correct statement over here. It is the wrong statement. Ribosomes are the site of protein synthesis. Of course, ribosomes are said to be as the protein factories, so hence they perform protein synthesis. It is the correct statement over here. Hence B is correct, A is wrong. Next one, middle lamella is the layer mainly of calcium carbonate. He says it is not made up of calcium carbonate, it is made up of calcium magnesium pectate itself. That's why this statement is also wrong over here, right? Option C is wrong. And the next one is, in animal cells, steroidal hormones are synthesized by smooth endoplasmic reticulum. So that's why here the statement is, of course, it is right one. Why? Because steroidal hormones are synthesized inside the smooth endoplasmic reticulum. That's why option B as well as option B are the correct answers. So A as well as C are the wrong statements. Hence what it shows is, so option A is wrong, C is wrong, B as well as D are correct. So choose the correct answer from your material itself, right? So these are some of the questions I have solved, and the next question is, who first saw and described a live cell? For the first time, a live cell was seen as well as described by one of the scientists, that is Leeuwenhoek. Is the first scientist, the one who saw that one, right? So microfilaments of eukaryotic cell made up of, already we know the endoskeletal system of the cell. What is it made up of? Microtubules it is made up of, microfilaments it is made up of, and such kind of microfilaments are usually made up of one of the proteins called as actin. Actin is the protein that makes the microfilaments over here, right? Microtubule is made up of tubulin protein. So microtubules are made up of tubulin protein, and microfilaments are made up of actin protein. Both together form the endoskeletal system of the cell. But albumin, globulin are one kind of globular proteinaceous structures, so fibrin. So but these are not making the microfilaments, so actin makes the microfilaments over here. That's why correct answer is option A, that is actin. Next question. In eubacteria, a cellular component that resembles eukaryotic cell is. See here, in eubacteria, one of the cellular component that resembles eukaryotic cell. Eubacteria itself is a prokaryotic cell, and eukaryotic cell it is compared with. So see the options: ribosomes, cell wall, plasma membrane, as well as nucleus. In all these options, so eubacteria, so how it resembles with eukaryotic cell. So bacteria have ribosomes, of course. So bacteria have 70s ribosomes, but eukaryotic cell has 80s ribosomes. That's why it is not so correct answer for this question. Okay. And the next one, cell wall of eukaryotic cell as well as eubacterial cell. So eubacteria, whenever you go through that moment, both are not resembling. Why? Because the chemical composition of the cell wall of bacteria itself is so murine or muramic acid, right? So in case of eukaryotic cell it is different. In case of cell wall, that is cellulose as well as pectin. So initially, so that's why this is also not exactly the same. See here, in case of prokaryotic bacterial cell and eukaryotic cell, plasma membrane whenever you consider, so this is resembling over here. So a lot of resemblance is seen in the plasma membrane, but so in case of nucleus, prokaryotic cell, that is eubacteria, doesn't have true nucleus, and eukaryotic cell has true nucleus. So that's why nucleus is also not confined to this one. The correct answer for this one is the plasma membrane. It resembles in both the eukaryotic as well as prokaryotic cell. That's why option C is the correct answer for this question. Next question is, structural element of chromatin is histone proteins, acid protein and DNA, nucleosome as well as nuclear matrix. See here, so what is the question? Structural element of chromatin is. So what makes the structure of chromatin material? Already I have told you, this is the nucleus. Inside the nucleus, what is present? The chromatin material is present over here. It is a coiled thread-like material lying inside the nucleus, and nucleolus is also present over here. And this chromatin material condenses, it goes on condensing itself so as to form very condensed structures called as chromosomes. The structure of the chromosome will be like this. So this is one chromosome. So this thread-like material makes this kind of chromosome. So by coiling of this chromatin material during cell division, so whenever you uncoil this material, whenever you uncoil this chromosomal thread, so this chromatin material is basically made up of some special structures I had explained in theory. So and they contain mainly the DNA as well as proteins. DNA as well as proteins together form one nucleosome. What it is called as nucleosome. See here, whenever the structure of this chromosome or the chromatin material is present, so the basic structure itself is made up of nucleosome. Hence, this model of DNA itself is called as nucleosome model of DNA. It is said to be as. Right, when you uncoil the chromatin material present inside the chromosome, so nucleosomes are found there. So they are the basic structures what makes this chromatin material over here. So the nucleosome, each nucleosome has the DNA. Each nucleosome has DNA, so associated with the protein, and those proteins are called as histone proteins. Histone proteins are present over there, and those histone proteins in theory clearly I have explained there are 4 types of histone proteins are there: H2A, H2B, H3 as well as H4. They are double in number, means they are totally 8 in number. Totally they are forming the octamer system, and around that octamer of the protein there coils the DNA around it, and that makes one structure. That basic structure itself is called as nucleosome. So clearly in theory we have discussed. So but in NCERT it is not mentioned, but here question arises. So for that reason this concept has been explained by me. So I hope it is very clear to you to solve this bit. That is, so structural element of chromatin is not histone, it is not acid protein as well as DNA, it is nucleosome. Nucleosome is the correct answer for this question. Nuclear matrix is not correct answer for this question. Nuclear matrix, as I said, so it is the matrix which is lying so very close to the nuclear membrane, inner membrane of the nucleus, right? So next one, glycocalyx is associated with, what it is associated with? So nucleolus, he says. Glycocalyx, so since it is confined to the prokaryotic cell, so there is no possibility of connection between what? So this glycocalyx as well as nucleolus over here. Second option is nucleus. So is there any possibility of the presence of this nucleus in prokaryotic cell? In prokaryotic cell, so especially whenever we were discussing the bacterial cell, that moment I had explained what is glycocalyx over there, right? So if it is one of the bacterial cell, so it's a capsule, everything, it makes the glycocalyx as I had explained, right? So thoroughly over there, this is glycocalyx. But in DNA, in this bacterial cell, the DNA itself is present, which is naked one. It is not so confined with a boundary, that is nuclear membrane is absent over here. That is why there is no matter of talking of this nucleus over here. That is why this is also not the correct answer for this one. Nucleolus is also not the correct answer for this one. Then, so plasma membrane. See here, so plasma membrane is there. So it is confined to, means glycocalyx is confined to the plasma membrane. So totally glycocalyx that makes the envelope of the bacterial cell. So in envelope, one of the component itself is what? plasma membrane. That's why plasma membrane itself is the correct answer for this question. But there is no matter of nucleosome over here. That's why, so answer for this question is option C, that is plasma membrane. I hope all these questions are very clear to you. Now, so next question. Chromosomes are composed of. What chromosomes are composed of? Already I have drawn the diagram of the chromosome over here, and this chromosome, since it is made up of, see here you can very clearly seeing over here that is DNA is present over here, along with that one histone proteins are also present there. Then chromosome is made up of DNA, histone proteins as well as, so on the DNA the synthesis of RNA also takes place. Very limited amount of RNA is also present over here in the chromosomes. Its percentage has been mentioned by me already, right? So DNA, proteins as well as RNA. Hence option, which option is correct over here? So that is option D, DNA, RNA as well as protein. So this is the correct answer for this question, right? And next question is, what is the next question? Which one of the following does not differ in E. coli as well as Chlamydomonas? So E. coli itself is one of the bacteria, means it is a prokaryotic cell, you must remember. Okay, E. coli bacteria itself is bacterial cell, that is prokaryotic cell, and Chlamydomonas, it is unicellular algae, right? So this unicellular algae itself is eukaryotic cell. So here in this question he is going to focus once again the difference between prokaryotic cell as well as eukaryotic cell. We'll have to identify the correct option over here, right? What are the options? Let us see over here. So ribosomes. Which one of the following does not differ? Does not differ, right? So one is ribosomes, option B, that is chromosomal organization, and option C, cell wall as well as plasma membrane. So means in prokaryotic cell as well as in eukaryotic cell both are similar, means in both the cells it is similar. The structural component of these cells is similar. So already we have solved this one in the previous bit, that is plasma membrane is similar among themselves. But in case of cell wall there is a difference. In case of chromosomal organization it is also different. In case of ribosomes it is also different. Why? Because ribosomes are 70s in case of prokaryotic cell and 80s in case of eukaryotic cell. It is also very clear to us. Then what is the correct answer for this one? Option D, that is plasma membrane. Come to the next question. Nuclear membrane is absent in. Nuclear membrane is always absent in prokaryotic cells. Then we will have to identify in the options which is the prokaryotic cell, right? So first option is Penicillium. Penicillium notatum is one of the fungus. What it is? So it is one of the fungus. So these fungi are said to be as achlorophyllous plants, since they do not consisting of the chlorophyll pigment but they have so plant like structure, right? Hence they are said to be as achlorophyllous plants. But so the cells of this fungi so are usually they are eukaryotic in nature. That's why so this is not confined to the nuclear membrane absence over here. So nuclear membrane is present in a eukaryotic cell. That's why this is not the correct answer for this question. Agaricus. Once again we came to the same fungi. Agaricus itself is one of the fungus. So this is also fungus. So that's why this is also eukaryotic in nature. So then Agaricus is also not the correct answer for this question. And Volvox itself is colonially colonial algae. So since it is colonial algae, algae are eukaryotic in nature, that's why it is also not confined to what? It is also not confined to, it is algae. It is also not confined to prokaryotic cell. And come to the nostoc, very important one. Nostoc, Anabina, Azolla. So all these are the best examples for blue-green algae. What are the examples? These are the examples for blue-green algae, BZA. So blue-green algae are prokaryotic in nature. They are basically prokaryotic in nature, and these prokaryotes do not have any nuclear membrane around the nucleus. Hence, the correct answer for this question is nostoc. Anabina is also best example. Azolla is also best example for these blue-green algae. So come to the next question. Very important question it is. Most of the hydrolytic enzymes of lysosome function at what pH they are actually functioning, right? So here the pH matters over here. What kind of pH? So already we know lysosomes are consisting of hydrolytic enzymes, and these hydrolytic enzymes are so acidic in nature. They are acting only in acidic nature, right? So basically they do not act in alkaline medium, neutral, or both B and C, right? So in these, so they are not acting, they are acting in acidic pH. That's why the first option A is the correct answer for this question answer, right? So, and let's go to the next question. 64th question, that is, see here, which is not true for active transport? Which is not true for active transport? What is active transport? It is the transportation of the material across the membrane against concentration gradients. Already I have explained what is concentration gradients. Everything I have explained. So there is a need of energy, a lot of energy is needed so as to push the molecules from the region of lower concentration level towards the higher concentration level through semi-permeable membrane itself, right? So this is nothing but active transport. The basic concept we know it better. What are the options given over here? We will analyze them one by one. Let us see the first one. It is a chemical process, he says. It's not a chemical process, right? It's not a chemical process. That's why this statement itself is not true. It is not true. So over here, then the answer itself is the first one. First statement is the answer for this question. And what is the second option? Let us see. Analyze all the options now, right? Required energy is obtained from ATP. Of course, it is 100% right. Why? Because so the energy is required for the movement of molecules from the region of lower concentration level towards the higher concentration level with the assistance of these ATPs. So these ATPs are nothing but energy packets. The energy is got from these one. Next option. It takes place through carrier molecules. Of course, carrier proteins. So through which the active transport that take place over here. So this is also the correct statement over here. It is not modified by enzymes. Of course, it is not modified by the enzymes. Okay, these all statements are right over here. This is the only statement which is wrong over here. That is why the correct answer for this question is it is a chemical process. Next question. Which one of the following structures is an organelle within organelle? So during the theory we have studied very well that organelles within organelles. Some of the organelles like mitochondria, chloroplast, what do they contain? Some other cell organelles they contain what? Ribosomes especially. Hence ribosomes are the smallest micromeres or microsomes, so which are present inside other organelles. They are said as organelles within the organelles. So then option A is the right answer for this question. Peroxisomes, they are meant for the metabolism of peroxides. So they are independently present in the cytoplasm, they are not present within other cell organelles. That's why these are not acting as cell organelles within the cell organelles. Next one is endoplasmic reticulum. It is an independent one, right? And mesosomes are nothing but the cell membrane in case of bacterial cell that makes what? The protuberance inside the cell, so that protuberance itself makes the mesosome, right? So these are all options are wrong, and the ribosomes is the correct option for this question. Next one. Which cellular part is correctly described? Cellular part is correctly described, right? So let us see the options first and let us analyze the options. Thylakoids. Thylakoids are flattened membranous sacs forming granum. Of course. So thylakoids are nothing but they are the flattened membranous sacs, secular structures forming the granum in the chloroplast, right? So that's why. So first one is the statement which is correct one. Next one, centrioles. Centrioles are responsible for formation of spindle fibers during cell division, right? Sites for active RNA synthesis, he is saying. So they are not the sites for active RNA synthesis over here. That's why statement B is wrong over here, right? Centrioles are responsible for the formation of microtubules which form the spindle fibers during cell division, to which the chromosomes are hanged and start to move towards both the poles. Already I have explained this one. Next one, ribosomes are said to be as protein factories. But what he says over here, those in the chloroplast are larger. He says ribosomes are larger cell organelles in case of chloroplast, he says, but actually they are not larger in case of chloroplast. They are smaller, that is about 70 S ribosomal subunits. So are present over here in case of chloroplast, but 80S is a larger one. That's why this statement is also wrong, right? Let us go to the next one. What is the next one? Lysosomes. Lysosomes are called as suicide bags of the cell. Let us see the statement regarding lysosomes. Optimally active at the pH of about 8.5. Usually these are acting at the high pH 1, 2, 3, 4 like this. So the total pH itself, pH scale is 1 to 14. So 1 to 6 itself is acidic one, 7 is neutral one, 8 to 14 itself is basic one. So but these are acting in 1 to 6 acidic nature. So right, high acidic nature is required for the action of these enzymes, right? So lysosomes optimally active. They are active at the pH of about 8.5 in the sense that, so it is more than 7, that is more than neutral condition. So this is basic nature. So in basic condition they do not act over here. They are not being active over here. That's why this statement is also wrong over here. Then which is the correct statement over here? So that is thylakoids, which are flattened membranous sacs forming granum. Right? Next question is 67th question. color of flower petals is due to? Already we have studied the different types of pigments over here, and especially we have studied in case of vacuoles, there some of the pigments are present over there, and those pigments are responsible for the color of the petals. There I had explained, right? So they are anthocyanin as well as anthocyanin are the pigments present, and they are responsible for the coloration of the petals, right? So let us see the options over here. One is xanthophylls, so carotenes, anthocyanin. Hence this is the correct answer for this question, right? They are the dissolved pigments within the vacuoles, especially it is present in the cell sap itself, right? Anthocyanin is the correct answer. Phycoerythrin is also not correct answer over here, right? Next question, and the last question here. Extranuclear DNA is found in. What is this extranuclear DNA? Extra means outside. Remember? So this is how we'll have to analyze the words over here. Extra means outside. Nuclear, except nucleus. The DNA present inside the nucleus, except that one, the DNA is found in. Means except the DNA of the nucleus, somewhere else what is present? DNA is present. Where is that DNA present? So out of this nuclear DNA. So that is nothing but, in case of prokaryotic cell, plasmids are nothing but extra chromosomal self-replicating DNAs, circular DNAs they are called as. They are also extra chromosomal in the sense that extra nuclear one. There is no nucleus, so that's why I said. So in case of eukaryotic cell, mitochondria, chloroplast, these are the membrane bound, double membrane bound cell organelles, right? So they consisting of their own DNA, RNA, proteins, synthesizing the factories called as ribosomes, right? So these can self-replicate themselves, right, independent or irrespective of the cell division, right? So that is why in mitochondria it is present, means DNA is present, which is not confined to, which is not related to the DNA of the nucleus, right? And chloroplast also the same, right? There it is also consisting of the DNA, which is the extra nuclear one. Hence, so let us see the options over here and analyze the answer over here. So find out the answer over here. One is vacuole. Vacuole doesn't have its DNA. Lysosomes, they do not have any DNA. They have only hydrolytic enzymes. Mitochondria has its own DNA, which is extra nuclear one. What about the Golgi complex? It doesn't have DNA, right? It has cistern tubules as well as vesicles, all together forming what? The Golgi complex. Dictyosomes they are also called as, right? So that's why correct option is option C, mitochondria. So these are the questions I have solved for you in this session. In the next session we will start with the next questions. Thank you.